So welcome everybody to the August Soils Network of Knowledge webinar. It's great to have a few new people attending today. So now I'd like to introduce Lawrie, who's presenting today's webinar at Soil Profiles and Soils of New South Wales. Roy is a soil scientist formerly with the Department of Primary Industries in New South Wales, and he's worked in many areas of soil science, including soil mapping and land classification, soil characterisation and sampling of research sites, and acid sulfate soil studies, land suitability for irrigation, land application for a range of organic wastes, the development of soil sampling machinery, and soil studies of archaeological sites. The work has taken him from the semi-arid inland to the high rainfall coastal areas of New South Wales. He currently works in communicating soils information, which he does extremely well, right? Many of you, when we've asked you what you would like as a webinar topic, have requested a general introduction on soils. And Abby Jenkins and I thought about who would be the best person in New South Wales that could cover that topic, and we thought of Roy, and he is ideal for it. I'll say finally that on a personal note, I met Roy several years ago at the Elizabeth MacArthur Ag Institute near Camden when I came to run a course on salinity for the Hawkesbury Nepean CMA. I had been told by the people down there that I should try to get Roy as a speaker, and it was fantastic that I did. Roy was so knowledgeable, but particularly so generous with his time and his keenness to share his knowledge on soils. So I'm very pleased to be introducing Roy today.
Thanks very much, Luke. Luke's asked me to talk about the features of New South Wales soil profiles. So today I'm going to speak about the main features of the soil profile and what they mean from the land management's perspective, using examples from across New South Wales, together with some questions that you might want to think about while you're looking at this set of pictures I'm going to show you of various soil profiles. First of all, I want people to be able to recognise the main features of the soil profile. What are the horizons or layers, and how are they derived? So the horizons are natural features derived by natural processes, and any changes to them arising from human activity often result in a different appearance of the soil. So there are features of the original profile in these two cores from Hyde Park. I want you to have a look at these soils here and see if you can recognise where the original A horizon is. The topsoil in the soil profile from the right, which is 3 metres long, is where the knife is over here. A very sharp boundary just there, and all that stuff on the top is not part of the profile, but it's all about human activity in the last century or so, what has happened to that particular soil. In another part of the park, that particular topsoil has been completely altered and obliterated by other things that have happened. So when you're looking at a soil, be aware that other people have been there before you. But using these soils, we can see that the original surface down here preserved quite well. Just here, the original topsoil, the A horizon. Underneath this is the yellow-brown B horizon, and down here is this pallid clay subsoil. The same pallid clay is visible on this profile over here, and underneath that is the shale that the soil was formed from. Next one will be, we'll talk about now the features of the A horizon. The natural surface layer, the A horizon, is characterised by a darker colour, and it's usually due to its elevated content of organic matter. This is derived from decayed plant residues. Organic matter acts as a glue, binding the soil mineral particles together into crumbs or aggregates, creating gaps or pores between the crumbs that allow entry of air and water. As you can see in this slide, this is a very well-structured A horizon. But even a big thunderstorm, it can give the ground a good soaking, but there is a limit to the capacity of water entry. We want that water to enter the profile, of course, but it's the A horizon which acts as the point where that actually happens. About 90% of the nutrients needed by plant growth are in the A horizon, and it's the most biologically active part of the profile. A fertile A horizon is needed by all plants, and this is the reason why most soil testing, when we take our samples off to the lab, is done on samples of the A horizon. Under pasture, we often see good root penetration through the A horizon, within and between the soil aggregates, and preservation of earthworm burrows, such as in this red-brown clay loam from the southern tablelands near Goulburn. It shows that the soil aggregates here are coherent and stable and allow good water entry. In the next photo shows a dark clay from a cultivation paddock in the Canamble area. This has had a history of minimum or zero tillage. By encouraging soil biology activity, the breakdown of crop residues, including roots, is more rapid. This recycles the nutrients that the crop residues contain, ready to be used by the next crop. On the other hand, you have a different type of soil in the south. This is the brown sand A horizon. Unless that has an abundant quantity of organic matter, sandy soils have no coherent aggregates at all. They contain few plant nutrients. They are permeable, they allow good root penetration, even after repeated cultivation. The profile on the left is many metres thick, with very thick sand cover in an old sand dune. The one on the right has only a shallow topsoil, half a metre thick, with a dense clay underneath. This clay subsoil is sometimes a benefit and sometimes it's a hindrance. The pale sandy A horizons are common in areas used for intensive horticulture, like this one near Richmond. Over 200 years of cultivation in this district, a seriously depleted soil organic matter levels, but sandy topsoils like this probably had little or no structure even at the time of settlement in the 1790s. Poultry manure is often used in the district to boost productivity, not only to supply nutrients, but also helping to raise water holding capacity. The other extreme, out in the west, this is another topsoil, which is completely structureless. It's a grey clay and it's got a smooth, hard surface. Most arid zone soils like this have very low content of organic matter. And unless the clay forms deep cracks when it dries out, their porosity and water entry, and consequently plant growth, as you can see in this picture, is minimal. The soil pores instead are filled with very fine clay particles produced during rainfall events. This process is often associated with an elevated exchangeable sodium content, and sodic soils, as they are called, are prone to this problem known as dispersion. In cultivated areas, gypsum can be applied to treat this problem. This is another approach used in the more well-watered areas, if you can describe it as that, further east. Extensive areas of bare ground on this scald can eventually be revegetated if there's enough soil moisture. Surface runoff is decreased by roughening the smooth surface of the scald or clay pan and then pushing up a low retaining bank around the edge where rainfall would otherwise run off. And in the top part of the photo here, you can see this bank over here and the rough surface over here. Unlike the Cutherrow site, there's even a few little bits of salt bush battling here, but we want a bit more cover than that. Landholders have successfully treated thousands of hectares of semi-arid grazing lands like this. Now we'll look below the surface and see how this affects plant growth. You'll notice in this this photo, this is a test for aggregate stability. Small soil aggregates, when immersed in non-saline water, show a range of behaviour. Stable aggregates stay intact on wetting, like these ones here. Aggregates of topsoil in well-watered coastal and tableland regions usually have enough organic matter to prevent breakdown, but their subsoil aggregates may not, and this is what happens to them. They disappear into a sort of a pool or spreading out of very small, very fine aggregates. The larger aggregates may collapse into smaller aggregates. This process is called slaking, and it also occurs widely and surface soils of the drier inland areas. Further breakdown into particles so fine that they never settle out, this is the process of dispersion I mentioned above, results in cloudy water all the time. All three behaviours can be seen in the one soil profile, depending on the particular soil horizon that is tested. So all these samples will come from different horizons. They've been from the same one, they're from the same one. Those two are from the same one, and probably that one too. You can see what's going on down here. Which horizon is stable in this particular profile at Young? That's a remarkable shot showing the stability of the A horizon over here with good vegetative cover, but very little vegetative cover down here. The B horizon, or the subsoil, is stable in this area, but the lower part is not. Obviously here you have a huge amount of slaking, the process where the larger crumbs break down into smaller ones, but in the lower part of the subsoil down here, it's more stable. And this part of the profile here is weathered granite, and it is more like rock rather than soil. This big ball shows you that plants obtain much of their moisture supply from the B horizon, especially long-lived, deep-rooted ones like this lucerne plant. Well-drained, porous B horizons are rare in New South Wales. Many of our subsoils look like this, I'm afraid, and they're full of dense clay that hold moisture tightly. Roots can then penetrate only if the subsoil's chemical and physical properties are favourable. Subsoil acidity is common in New South Wales, but not in this southern riverina soil. Like many of our inland soils, its subsoil is alkaline. Do soil horizons affect plant growth? Well, it depends on the plant as well as the soil properties. Subsoil compaction produced by passage of heavy farm machinery is often first noticed by its effect on plant growth and is not easily repaired. The other question you might like to think about looking at this root slide is how much of these plant roots survive after we harvest the top? What actually helps in breaking down these roots and recycling their nutrients back to plant available forms? Now we'll look at features of the whole profile. We've finished looking at subsoils. These soil profiles from the Shoalhaven floodplain occur on a dairy farm with irrigated pastures. Each has a different amount of RAW, or readily available water, about half of the amount of plant available water that can potentially be provided by the soil. The main factor influencing this water supply is the texture and the thickness of the A horizon. So the soil on the left over here has a very nice thick A horizon like this. I think I'll have another few more million acres of this, please. But these soils over here have a relatively thinner A horizon. So the water storage is limited by the capacity of this clay in the B horizon to supply it. You can see there's a difference across the farm. As in most places in New South Wales, we have a big range of soil properties with any particular land holding. We all have to learn to live with this. This is shown very well in the next slide with the soils from the floodplain of the Macquarie River from a property at Mundry Bar. There's a wide range of features, some of which have been modified by land use. There's a brown and a grey cracking clay These were considered too difficult to cultivate until the 1970s, and they were rarely used for cultivation and mainly for dryland grazing. But these are now the favoured soils in the district for irrigation. This is because the water enters the soil very rapidly through deep cracks when the profile dries out. They are slow to drain after rain. They're prone to subsoil compaction at harvest time, even though the surface has dried out. This one here, this is a brown loam over a red clay, the favoured soil for dryland cropping for over 100 years. But this particular A horizon has lost much of its organic matter. You can see it's just formed in a big chunk here. It is now virtually structureless. Looks like a bit like a lump of concrete, if I can find my arrow. Yep, there it is there. The red B horizon underneath has a moderate structure and good water holding capacity for any moisture that finds its way through this A horizon, which has been damaged by cultivation. The uncultivated soil next to it, number 4, is used only for grazing. This still has plenty of organic matter left in the topsoil. Just have a look how dark that A horizon is. And the amount of organic matter gradually falls away with depth, resting on an ancient alluvial clay underneath down here with reasonable capacity to store moisture. Look at the contrast in the subsoils here, even the contrast in their surface soils, all on the one farm. Those hard-setting loamy surface soils with a history of dryland cropping can be deep ripped to improve moisture penetration, but the improvement this offers may only be short-lived. Adding extra organic matter is a medium to long-term strategy aiming to improve that soil structure. Conservation tillage practices, including controlled traffic and tram lines, would help increase soil biological activity. If you can't do that, have a look at the implement used here and the massive amount of engineering behind this and the potential cost involved. Soil acidity and alkalinity. Field pH testing. Very useful for deciding which soil to sample for lab testing, as well as checking for major limitations imposed by soil chemical properties. Test results can indicate just how different or how similar chemical properties are between soil horizons. Even from the one paddock here is a red soil here, which is a little bit acidic, shown by the yellow colour on the surface, gradually becoming less acidic with depth. Down in the bottom of the paddock, it's neutral pH, and it gradually becomes purple down the bottom here, indicating it's alkaline in the subsoil. How can you pick a saline soil? Well, apologies for the next slide, which is from South Australia, because salinity is rarely visible in the soil profile in New South Wales, though the presence of plant roots is one indicator of the absence of salts. If you can see a plant root that's not likely to be too salty unless it's a salt bush. Surface vegetation is another indicator as well. But which areas on this particular farm in South Australia are salty? The average rainfall here, by the way, is about 500 millimetres. How far down is the salt in the paddock surrounding this? Lab tests are the best guide to give you an indicator. It's very hard to see this in the profile. Dryland and salinity in New South Wales can exist even in coastal regions where rainfall is below 750 millimetres. Some viewers will recognise these soils. They're from Menangle, just outside of Sydney. The annual rainfall here is below 800 millimetres. Are these soils salty? It's impossible to know, but they have clear horizons, and you can see easily the A, the B and the C horizon. The darker topsoil by organic matter, the red coloured B, the horizon which has the maximum colour intensity over here, and the C horizon where you're approaching the weathered rock down here, but it's still not rock. Each of these horizons, two horizons, have different chemical properties. This one here is immediately downslope of that one. But this difference in chemical properties, did it matter for Elizabeth MacArthur or the other early dryland grazers? Here she is over here, thinking about what to do with the sheep. These soils now support intensive dairy production, however, and then dryland cropping was also important here for 50 years. And what was the likely impact of these land uses on the soils? What did it do to the topsoil? We might like to think about that. This is a red chromosol, the proposed state soil for New South Wales. It has a weakly structured A horizon of a sandy clay loam resting over a medium clay B horizon, which is only moderately aggregated. But what would be suitable management practices for dryland cropping on this soil? When James Roos farmed the soil in the 1790s, what did he actually do? You might think about why the first wheat crops failed in Sydney, but not on this particular profile. The soil profile at Hyde Park might deserve another look. Were the soil properties to blame for starvation in the colony or not? Perhaps it was the lack of agronomic expertise. So I'd like you to think about soil management and these different soils of New South Wales we've all had a look at. And I'll open the talk up now for questions. I think I'll hand it back to you, Luke.
Thanks, Roy. Fantastic introduction to soils around New South Wales, finishing up with Hyde Park and Parramatta. I like that. I just might ask Roy, might take the liberty of asking the first question myself. Now that I see those monoliths, I'm aware, and I've been with you when you've done an amazing job of digging up a lot of soils around New South Wales, and I'm aware that, you know, some of them still reside with us. Do you want to just make a comment about them as a resource and how you'd like to see them used?
Yeah, look, these monoliths work best when you have a photograph of the site and the district around it, and some details about land use and why the monolith is important. Little boxes with a description of the profile next to them are also useful, but you can use this to put a particular message out there that you might want to use in this particular soil for. For example, this red chromosol could be used to talk about dryland cropping management. It could be used to talk about carbon sequestration. You could be talking about a lot of different land management messages you might want to put out there, but you've got to draw people's attention to those and how the soil affects those land management practices.
Thanks, Roy. Gavin Tinning up at Wallumborough has a question.
Yeah, interesting presentation, Roy. Thank you. I just had a more general question across soil types. Is that in terms of irrigation or watering, what sort of signs tell you whether your soil is sufficiently irrigated or well watered? Are there, other than going to the step of having, you know, technical equipment, are there other, you know, symptoms or signs that'll show that you're irrigating your soil sufficiently for the crop?
Yeah, in the you can use visual indicators. Looking at the topsoil, the colour of the topsoil, if it darkens in colour when you add moisture to it, in other words, you put a few drops of water on the soil and the colour changes and it goes dark, that soil is too dry and it needs to be irrigated straight away. If it doesn't go darker in colour and stays the same colour, means there's a little bit of moisture in there. And if it's a loamy soil and you can roll that soil into a rod and then without it breaking, it's probably a little bit too wet. But if it breaks into lumps, it's probably okay as far as the moisture level is concerned. Yeah, but it's starting to dry out a little bit. Obviously in a sandy soil you can't make a rod. In a clay soil, if you can make a rod and bend it round into a ring, it's obviously way too wet and doesn't need to be irrigated.
Thank you.
Very practical answer, Roy. That's and really helpful for people. Douglas Fox.
Yes, my interest in soils is the fact that soils are a living entity, not just a medium to stand a plant in, and they are the home for microbial and insect life. But this has been avoided so far.
I Did you remember, Douglas, I showed a photo of a canamble soil with termite larvae sitting in it?
Yeah, and there was one wormhole also on another slide.
Yeah, that's right. Yeah, here we are, that place. Yeah, these homes that insects and other, soil fauna make in our soils, very easily damaged by cultivation, of course. But the important thing that they do for us is to help recycle plant residues in the soil, converting them from the non-available forms when they're actually, in dead plants into usable nutrients by the next crop. This particular farmer doesn't use any fertiliser. He incorporates all these residues into the soil. And you saw how a lot of plants have massive root systems below the surface, which are just ignored by most people.
Yes, in Australia, soil microbial life is just not considered overseas. It's highly valued, and they feed the soil microbes to grow a plant with.
Yeah, well, you can see that these plant roots here have got enormous proportion of the total biomass is below the surface, and we need those microbes in there to break down these plant residues, these subsurface residues, to recycle these nutrients, and also these this recycling process is assisted though by temperature and moisture. If we have very dry conditions in the soil, the microbes are going to fall asleep and not do much work for us, and the same goes if they're too wet or too cold or too hot. So microbes need the favourable conditions to do the job for us, and a lot of these conditions are not well. The weather is sometimes not very helpful, put it that way. So there is a lot of variation in microbial activity in Australian soils associated with fluctuations in soil temperature and moisture. So it's very difficult to get a quantitative test on how much microbial activity is actually present in the soil at any particular time because of the fluctuations in soil, moisture and temperature conditions. So when we did have a test which said there was so much microbial activity, how would we know if that was enough or not? We need lots of trials around the place to work out if that number is high or low or in between for that particular site. There's a lot of work needed, in other words. But we do have farmers who want to improve things, and there's a lot of experimentation going on out there because landholders are very interested in this. This was sparked particularly by the skyrocketing fertiliser prices about 7 or 8 years ago in 2008, when the price of fertiliser went through the roof and people wanted to investigate alternatives.
Yeah, thank you very much, man.
So moving on to the next questions from Nev Rees, Roy. Now Nev says he's sorry he doesn't have a mic, so I'm going to read Nev's question out. And his question is, can you please give a brief overview on how carbon sequestration is tested in soils?
Yeah, thanks for your question, Nev. There's several tests that we use to measure soil carbon levels, and there's research in progress trying to work out faster and more rapid ways, easier ways to do this. But unfortunately at the moment, a chemical test in a laboratory is the only way that seems to guarantee some success here. What we do know is that the rate of change of carbon levels in the soil is quite a slow process, so we're trying to measure very small changes in this level, and this is a difficult job because of the variation, the spot variation in carbon levels across the paddock. You can get big variations because of the different patterns of vegetation across the paddock and the different degrees of breakdown of the plant residues into different types of residues. So that means that when we do a soil test for carbon, we've got to take a number of samples, and more is better, in fact, and make a composite across the paddock. And then you can get a rough idea of what's happening across the whole paddock. So 30, even 40 samples are sometimes necessary to do this. And if you want to do that at more than one depth, there's another big job for you, right? Any sampling below the surface, even 20 centimetres below the surface, is normally done using a mechanised drill rig. Cost comes into the frame again to do all of that test, and so it's a slow, tedious and expensive process at the moment. If people can come up with another way of measuring and assessing changes to soil carbon levels, then we'll be able to what can we say, give a level of give a bit more assistance to people who want to do this successfully. Where it stands at the moment, we're not able to do this readily, and most of the literature in the scientific literature which is attempted to measure soil carbon down the profile is affected by these measurement difficulties, and so people tend to concentrate on very small plots where they can monitor these small changes more rapidly. And what I'm talking about rapidly here is you might need a trial that extends over 10 years to do this.
Yeah. So I'm now moving on to the next question, which comes from Andy Taylor. Andy asks, have you seen soil benefits through practices like pasture cropping and planned grazing management?
Oh, thanks for your question, Andy. Yeah, thanks to a fella called John Lawrie, a bloke with the same surname as me, but absolutely no relation, I want to tell you. I visited a pasture cropper in the area south of Coonabarabran a few years ago, and I was able to have a look at this process there. This was a program which was on his place being carried out on a rather sandy soil. It had very little or no structure, but the aim of the project they seemed to be getting some results, some favourable results out of that. I think that one of the things that we can learn from these projects is that you have to look after the structure of our surface soils better and improve our aggregation, improve water entry, improve biological activity as a result. As I said before, when our soils are too dry because they haven't allowed enough water to enter because of damage by cultivation, then the bugs don't get a chance. They don't get a look in because there's not enough water there for them to do their job. But if anyone can work out a way how to farm soils without destroying the habitats of our soil fauna, well, we'll all be, especially those of us who have concerns about, the low rainfall parts of the world, we'll be able to see some opportunities, to improve these, rather delicate soils that are easily damaged by cultivation. And I'm following with interest the progress of these people who want to do pasture cropping. I have met some people who have done this, but haven't had a lot of time to look at the differences across the farms between the areas that have been cropped and those that haven't been used in this way. The changes to grazing management are very difficult to see in the soil profile. The best way that I remember looking at these, after visiting one farm that's only done that, and I've had limited experience in this regard, was when I had a spade with me, and I walked around the farm on a spade, and the areas where the grazing management had improved soil structure, you could actually push the spade into the ground. It hadn't been compacted to as much as extent. You had better plant growth, better moisture penetration. You could actually push the spade in other areas which hadn't been managed quite so successfully. The spade would hardly go into the soil at all. It was just too hard, and there was a lack of aggregation. And this, but the grazing management changes are very, very subtle, and that's changes to soil properties are brought about, I think, mostly by the changes to the way that pasture grows in those different grazing practices. I hope that helps you a bit.
Thanks. I'll just add to that, Roy, that another person who's done some terrific work on pasture cropping and the soils in it is Warwick Badgery at Orange. So if you know Warwick, you could look up his work there. And I've got a comment here, Dave Mitchell.
Yeah, just a comment on the so the grazing trial that we ran at Panuara here in central New South Wales was based on looking at different grazing treatments and the subsequent effect on productivity as well as some NRM outcomes. We measured soil water, moisture and a range of indicators across that experiment for about 5 years, and there was no change in soil. We could not detect any difference between soil moisture content across grazing treatments, and we did a number of physical soil measurements to look at see if we could detect any changes, and the major one was just looking at the changes to infiltration under different grazing treatments, and we couldn't detect any. That was, we were using rainfall simulation to measure that to those changes. So that was just a quick comment there, Luke.
We've now got a question from Helen Wheeler. Helen says, need to measure total organic carbon because inorganic is too labile, question mark?
Okay. Inorganic carbon, that's a nice thing that we often see present naturally in soils as calcium carbonate, which is lime. A lot of our drier inland soils have lime, some of them even have lime right to the surface. This stuff is formed naturally in the soil out of the soil minerals and hasn't got anything to do with plant growth or organic matter. It's formed independently of organic matter. That's why people don't usually measure it. In this, you can have huge accumulations of lime in the soil, of course, great big lumps or concretions. Some of it is like rocks, and some looks like old concrete, some are like dinosaur bones, isn't it? But it's a subsoil thing, and but sometimes this lime occurs as very, very, very fine particles, too fine to see, detectable only when you drop a little bit of acid on the soil and it froths and fizzes. And this is when this reaction is called, we call it lime present in the fine earth of the soil. You just can't see it, but you can only tell it's there when you do this test. Obviously, it doesn't occur in acid soils because the acid, natural acids produced by organic matter in the soils they've dissolved it all the way. And it's just not there anymore. So when we do a test for carbon in soils, we always measure organic carbon, which is present in the soil regardless of what the acidity or alkalinity of the soil may be. And to do this, the commonly used test is based on heating the soil up, just cooking it in a stove if you like, and burning it off, because a lot of that carbon just catches fire, right temperature, and up she goes in a puff of smoke. And the other way we do it is we attack that organic carbon with hydrogen peroxide, and that oxidizes it in much the same way. And then we can tell how much we've lost by how much carbon moves off after it's oxidized through to carbon dioxide by cooking it up with a bit of hydrogen peroxide. They're the common methods that are used. Does that help you a bit, Helen? But there are other methods that are being tested by various research programs around the place, internationally too, trying to work out a good way to do this.
Great, Roy. Yeah. Carol Rose has got a question.
Hi, Roy. Can you hear me?
You bet I can, Carol. Lovely to hear from you.
Yeah, I was wondering a few years ago, we had lots and lots of work done on the coast about inland, about acid sulfate soils. I was wondering how much inland acid sulfate soils has been found in New South Wales.
The short answer to that is not a lot.
Is that because we haven't been looking, or?
Yeah, that'll be part of the reason. Some people have been, lifting up rocks and stuff to see whether it's what's lying underneath, but there hasn't been a need to go out and chase for this because we haven't had any reports from landholders that they've seen anything like this. That'd be probably the first thing I'd say about it. In South Australia, the land of salt, well, there has been a lot more found out there, and I think there's sort of geological reasons for this that maybe we don't have in the same sort of situations happening here in New South Wales, but I can imagine that some of these South Australian situations could exist in New South Wales, but they would be in small, confined areas. So we would probably see them as changes to the vegetation. That would be the only way I could think about it. But they'd also have to be in wet spots, because you have to have waterlogging of the soil to favour the formation of the sulfide minerals. And if you don't have waterlogging and the soil's got contact with the air, you will not get sulfides formed in the soil. But you could have them if the sulfide minerals were there formed by geological processes rather than soil bacteria, which is a way that's formed in coastal soils.
Yep, Roy, I got a call from someone up at Ralston Way in one of those deep sandstone valleys, and they had a very low pH in a peaty area in one of the
Yeah, that's all right. Yep, they're allowed to have that. Yeah, because these very low pHs in peaty areas are caused by production of organic acids. Because you've got a lot of organic matter in a peat, a lot of production of acids if you've got the right temperature and moisture regime, and therefore you've got acidification going on that way. And people shouldn't get too excited over these things, because these naturally acid peats are capable of producing pHs below 4. But there's not necessarily any sulfide minerals formed, which is what you need for acid sulfate soils. So you've got to actually have black, smelly soils. The way that I have seen these inland soils go sometimes, if there is enough sulfur in the soil and by natural situations, and you have temporary waterlogging produced by a flood or something like this, the pasture will go black in puddles, around the place. And this black colour occurs, you've seen it on the coast too, I think, Carol, where you get associated with black water events, And I think there's nothing stopping the same process happening in inland areas, provided there is a high background level of sulphur in the soil. The KCl40 test, however, generally indicates that a lot of our inland soils have got very low sulphur levels, and well, we don't see that on the coast. And a lot of our coastal soils, we've got 10 times or 100 times as much sulphur on the KCl40 test than we have in our inland areas. So these coastal soils are sort of already, primed, if you like, to generate black water. But not too many of our inland soils are like that. So that'll be my thinking about the Ralston place. If you had a sulphur test, a KCl40 test there, that would, and it was the answer was in single digits. It's definitely not acid sulfate.
Now, moving on, Helen Wheeler, where you worked on the Crookhaven River floodplain for PASS/ASS in a wetland behind a 1970s levee with no floodgate.
Yeah.
Would it be possible to form a modern 50-year-old, this is capital letters PASS, sorry for the site soils,
sure.
and you'll explain what PASS stands for. Site soils are always
I know what that is. That's okay. I know what it is. Yep.
Yeah. You might explain for our audience. Site soils are always wet, sometimes inundated by rainfall. What tests would you recommend, Roy, to check for PASS on these sites?
Well, I haven't touched on this area in the seminar. Of course, it's a whole new subject. You've lifted up a corner of the carpet here, and now you expose some of the nasties that are sort of underneath. PASS stands for potential acid sulfate soil, and these potential acid sulfate soils are formed under waterlogging conditions, and you have to have that. If you don't have any waterlogging, and you've got to have an absence of oxygen produced by waterlogging, you will not get the soil. But of course, if you have a waterlogged soil and it stays waterlogged for a very long period of time, and it doesn't have any oxygen dissolved in the water, and how does that occur? That occurs because you've got a lot of organic matter dissolved in there. So what happens with, for example, if in that situation where you've got a coastal situation behind a levee bank like that, if water hangs around in a corner in a low spot behind that levee bank and it's had pasture growing on it, the pasture will die because of the waterlogging, and then the bugs will want to attack that dead pasture and digest it. And first of all, they'll use up all the oxygen that might be dissolved in the water, and those oxygen requiring bugs, well, when they've finished their job, there still might be quite a lot of pasture left over, and the anaerobic bacteria that don't need oxygen will kick in. They'll be lining up for their turn at the table as well, and they get their the oxygen they do need for breakdown of that organic residue out of the sulfate that's present in the soil. And sulfate, iron is sulfur combined with 4 oxygen atoms, and so they break down the sulfate and convert it to sulfide, which has no oxygen with it. And so it's a reducing process. In other words, it's called a way of sucking oxygen out of the chemical rather than out of the air. All right, so once you've done that, you've got sulfides floating around in the soil water. And one of the other things that happens in soil and in waters which have no oxygen in them is that the iron minerals in the soil are dissolved. Normally in contact with the air, the iron minerals are not dissolved. They come out and they react and they form iron oxides of various types, and they give a rusty iron stain around to the water. But in reducing conditions, they're dissolved and you don't get any stains. In fact, you can have beautiful clear water. But under that situation, yeah, you're getting sulfides formed in the soil, and in these coastal floodplains, you don't get beautiful clear water. You generally get it goes all black and stinky. And under that situation, you're getting the formation of potential acid sulfate soil minerals at the soil surface. Right at the soil surface. As soon as that soil surface, and the water, by the way, in these puddles at the soil surface, in the potential acid sulfate soils, is not acidic. It's not. It's perfectly okay. Surprise, surprise. But when you expose it, the minerals that are formed underneath it, the black sulfide minerals, most of them contain a lot of iron, to the air, they are attacked by the oxygen in the air. They release sulfuric acid when the minerals break down, and that makes any subsequent water that might land on it as acid as anything. And then you can get very strong acidity conditions which will kill vegetation and kill fish if that water gets back into the river. This is a process which happens naturally in lots of coastal areas anyway. You don't need to have embankments and that, because coastal floodplains are not perfectly flat, so there's natural depressions in the landscape where this process happens. Normally the volume of this water in natural conditions that's produced by flood events is not that big, and these drainage lines, the natural drainage lines that feed this acid water back into the river are very puny, and so the flow is quite gentle. But when we build big drains and big levee banks all over the place, we potentially can increase the area that becomes sulfidic, if you like, turns black, and then after, when it dries out, it becomes oxidized, it generates acid. So the answer to your question about can the levee bank in the 1970s, do this, it will just help to enhance the natural processes. If the levee bank is actually stopping the area from draining properly, which I have seen on the northern Shoalhaven floodplain, yes, they do. It's not a deliberate thing, because the levee bank is actually, in this the case that I'm aware of, was produced by people cleaning out the drain and dumping the spoil on the land next to the drain, acting as a levee bank, and so water couldn't reach the surface of the drain anyway. This was an inadvertent result, and so there was water piled up at the back. All you need to do in that situation is to dig a few breaks in the levee bank so the surface water can drain away when you get some flooding. I hope that sort of fleshes out the process a little bit more, and certainly the Shoalhaven is a really interesting area for people who want to go into this area of research, and it's a whole new subject, but many of our coastal rivers experience similar problems like this.
Okay, Roy. Yeah, I've got a comment back here from Helen, who doesn't have a microphone. Before I read her comment, she had two questions she asked there. What tests would you recommend to check for PASS on these sites? Do you want to quickly answer that, and then I'll read your comment?
Yeah, okay. Yeah, go to the swimming pool shop, the swimming pool chemical suppliers in Nowra, buy yourself a bottle of hydrogen peroxide, the most concentrated stuff you can get. And walk around your surface soil and with a little dropper and drop some of that peroxide on the ground and see if it fizzes. If the soil surface is black and soggy, they're the places to do it in. It won't fizz if you see any well aerated or dry soil, because the black stuff is not stable when it reacts with the air. But it's unstable and quite unstable then, and there's a fizz test you can do. You have to know though that there's other things in the soil that can make hydrogen peroxide fizz, and that is organic matter itself. Soil organic matter, generally, when you put the sample of soil, say you put some in say in an empty Vegemite jar, and put a bit of your peroxide in the Vegemite jar, you'll get a head on a beer, like a froth, a nice light froth. That's a normal reaction. Any soil with no potential acid sulfate material will produce a light froth if it's got some organic matter in it with peroxide. But when you have some sulfide minerals present, you'll get a volcanic reaction, and then gases will come out, and it'll start puffing and chuffing like a steam train. There's quite a significant difference in the visible reaction. That's one way to do it. The other way to do it, of course, is to take a sample of your soil and send it off to this lab at Southern Cross Uni or a North Coast way, and where they'll give you a proper acid sulfate test, and will give you they'll do a test there. It's called the chromium reducible test, which is reduced inorganic sulfur, and you'll be able to put a number to that. And if you want the ability to put a number to how much sulfur you got in there, will be a guide to how much lime you need to apply to neutralise any acid that might be produced by that area if it dries out. And farmers who do, and other people who want to fix acid sulfate soils have done this test, and it's nothing to be afraid of. The acid sulfates are not to be, they can be managed now. There's ways around it, and there's guidelines on how to manage this. So they look a bit fearful and lengthy, but it's a case of, if you know what you're doing, then the problem, and you've got deep pockets, mind you, provided you don't have to treat, you know, thousands of square metres or thousands of tonnes of soil, well, it's not a huge problem. But if you're in the construction business and you're building, you know, highway bypasses around Gerringong, what's happening at the moment, you might dig up some of this stuff.
I think we couldn't have got a better person to, you know, soil generalist with not just a generalist, Roy, but the depth of your knowledge is quite phenomenal, and love the way that you explain things too, the frothing like on the top of a beer. That just helps people understand these things, and you can get your own hydrogen peroxide. So thank you so much, Roy.
All right.
And we'll talk to you soon. And thank you everybody for attending. So thanks everybody.